mitotic phase, or otherwise we can call it as division phase. So here this is a phase where the nucleus will divide to form into two daughter nuclei, and also the cell will divide to form into two daughter cells. So here it is starting with the first stage, karyokinesis. Karyos means nucleus, kinesis means division. So here in the karyokinesis, the nucleus will divide to form into two daughter nuclei. So whatever the condition of the nucleus that is present in the parental cell, so the same quantity and quality of the chromosomes are maintained in the daughter nuclei also. So likewise here, one parent nucleus is divided to form into two daughter nuclei in the karyokinesis process. And for the conveniency, the entire process of karyokinesis was divided into prophase, metaphase, anaphase and telophase. So these are further small changes can be observed in each of the substages. So let us discuss the process starting with the first stage, that is prophase. So prophase actually in the interphase. So if you see the interphase nucleus, the cell will be like this. With the nucleus, that is nuclear membrane, and the chromatin is irregularly present with a network like structure. This is a cytoplasm. This forms a cytoplasm, cell wall. So inside which we can see one more layer, that is cell membrane, that is plasma membrane is present. This is a nuclear membrane. In the nuclear membrane, this is a nucleoplasm. So this is a chromatin. So here the chromatin is long elongated thread-like structure present during the interphase stage. So here and there we can see one or two or sometimes more than one spherical shaped darkly stained structures called as nucleolus. Now when the cell enter into the prophase, so we can observe a change in the nature of the chromosomes, that is condensation of the chromosomes will take place, so that the long thread thin structures of chromatin are converted into rod shaped thick structures, those are called as a chromosomes. So here as it is the first stage, pro means first, phase means stage, the first stage of the division stage, that is why we call it as a prophase. So here the prophase is divided into we can say that the changes that are taking place during the prophase are categorized into three stages. So those are early prophase, mid prophase and late prophase. So in the early prophase, the chromatin which is present, so here gradually the coiling will start. Coiling of the chromatin will be taking place. So due to the coiling of the chromatin or the condensation process, we call it as, we can see a change in the structure of the chromatins which from that of the interphase stage. So this is a cell with a cell wall and the plasma membrane inside it. So this is a nuclear membrane. So here inside this one we can slightly observe that the small structures will be formed like this. And here and there we can see the nucleolus also. This type of small structures will be formed due to the condensation of the chromatin material. So likewise here we can see a definite structure related to the short and rod shaped structures can be formed during the early prophase, where the condensation of chromosomes will be taking place. And here in the mid prophase, the chromosomes will be converted into the perfect rod shaped structures. Nucleolus will also be clearly seen here. The perfect rod shaped structures can be seen. Here in this stage we can clearly see the presence of the chromatids, which are attached on its longitudinal part at one middle point. That middle point is called as a centromere. So these are the chromatids. These chromatids appears to be exactly attached at a central point called as a centromere. Sometimes they may attach not at the exactly center point, but wherever the point of attachment is present, that we call it as a centromeres. So this type of perfect chromosome organization can be observed during the mid prophase of the prophase stages. Then during the late prophase, the further changes which are taking place. So here, before going to the late prophase, here we can clearly differentiate the cytoplasm from that of the nucleoplasm. Here also cytoplasm is very much clearly differentiated from the cytoplasm because of the nuclear membrane. But here in the late prophase, the nuclear membrane gradually disappears. So we can show the diagram like this with a cell. The plasma membrane inside the cell wall, and here we can say that a dotted structure is represented here. This dotted structure represents the disappearing nuclear membrane. Disappearing nuclear membrane, so that by the time of completion of prophase, the nuclear membrane will completely disappear, so that the nucleoplasm and nuclear membrane, we can't differentiate them. Both of them are mixed with each other, and inside which we can clearly see the presence of the centromere containing chromosomes with definite rod shaped structures. And of course here the disappearance of nuclear membrane and also the disappearance of nucleoli can also be observed here, or nucleolus will also gradually disappears. Apart from this one, the endoplasmic reticulum and the Golgi complex also disappears, or Golgi apparatus will also disappears during by the end of prophase. And in the early prophase, so we have seen that the chromatin start coiling and condensation. Apart from that one, the viscosity of the cytoplasm increases. So this type of changes are taking place during the entire process of prophase, and by the end of this prophase, so we can see that all the chromosomes are released into the cytoplasms, where we cannot differentiate the nucleoplasm with that of the cytoplasm, in which the chromosomes are distributed irregularly throughout the cytoplasmic contents. And apart from this one, the spindle fibers which are required for the formation of the spindle apparatus in the plant cells, and the astral fibers which are formed in the animal cells, all are being initiated during the prophase stage only. In animal cells, here we know that the astral fibers will be formed, which have been formed by the centrioles and centrosomes. Centriole and the centrosome. So here two astral fibers are formed, so we call them as amphiastral. Amphiastral spindle will be formed here. So here these are they will come and attach to the mid of the centromeres during the metaphase stage. Then in animal cells that type of change is observed, but in the plant cells, so astrals are not formed. Astrals are not formed, so we call them as anastral fibers. But here these fibers are made up of the microtubules, and which is made up of a protein called as tubulin protein. And here all these are initiated during the prophase stage only. That's why we say that the initiation of spindle fibers will start during the prophase, but the completion of the spindle fibers will be taking place during the metaphase of the cell division or mitotic phase. The second stage in the mitotic phase is metaphase. So this is a stage where the orientation of the chromosomes in the equatorial region that is all the chromosomes will come to the center of the cell, and this is first change. The second one other change is the formation of the spindle apparatus. So these are the two major changes which are taking place during the metaphase. Because of these two changes, all the chromosomes will come and lie on the equatorial plate, which is formed in the equatorial region of the cell. So during this one, so here as we are discussed in the animal cells, the amphiastral spindle fibers are formed. That is, amphi means two and astral means star shaped. So two star shaped spindle structures are formed, whereas in the plant cells there is no astral, so it is formed by the microtubules and the microfilaments. So these microtubules and microfilaments are made up of a protein called as tubulin protein. Major quantity is made up of the tubulin protein. Apart from that one, the actin fiber is also been present. So here nearly 90 to 95% of the spindle fibers is made up of proteins, and 3.5 to 5% is made up of RNA molecule, and traces of other elements like lipids and some other organic substances are also been there. But the major constant part is a protein. In the proteins also the major type of protein is a tubulin protein, and actin and myosin protein is also been there, so to the little extent. So likewise the spindle fibers which are spindle shaped, the spindle shaped fibers are formed in the cell, which will converge towards the poles. Converge towards the opposite poles. So as they are converging towards the two opposite poles, so we call it as a bipolar spindle apparatus. Bipolar spindle apparatus. And here this bipolar spindle apparatus is made up of the spindle fibers. So nearly each fiber is made up of 4 to 20 microtubules. These microtubules are joined with each other to form a long and elongated structure called as fibers. So here these fibers which are forming from the two opposite poles, they may sometimes attach with each other to form a continuous fiber. So like continuous fibers are formed, and sometimes discontinuous fibers are also been formed. The continuous fibers means the fibers which are formed from the two opposite poles, where they will fuse with one another to appears to be forming from one pole to the extending up to opposite pole. So those are called as continuous fibers. But there are some fibers which are not continuous with each other. Such type of fibers are called as discontinuous fibers. So these are in the form of chromosomal fibers and interzonal fibers. chromosomal fibers means the fibers which are arising from the spindles. So, spindle apparatus that will come and attach to the kinetochore region. Kinetochore region, that is the centromere part, the kinetochores which are present in the centromere. So, that is the reason why these are called as the chromosomal fibers. Interzonal fibers are the fibers which are extending from the poles. So, they will not join to the opposite formed fibers, but some fragments of these spindle fibers are remained in the equatorial region. Those are called as a interzonal fibers. So, here the chromosomes are attached because of the chromosomal fibers and also the interzonal fibers. Mainly because of the chromosomal fibers, as chromosomes are organized on the equatorial plate in the equatorial region. And the spindle fibers are having negative charge towards the poles and positive charge towards the center. Consider the cell. Like which is having the cell wall, plasma membrane, here the spindles which have been formed. Like this, they will reach up to the center. Where they will come and attach to the centromere of the chromosome. So, this is a spindle apparatus. The spindle fibers, and each one is a microtubule. Likewise, each fiber is made up of 4 to 20 microtubules. So, here the spindle fibers will come and attach to the centromere, so that the tightening of the centromere will be taken, tightening of the chromosomes will take place. Because of that one, all the centromeres appearing to be present on imaginary line. So, this is called as a equatorial plate, or otherwise as it is formed during the metaphase. So, we call it as a metaphasic plate. And as this plate-like structure is formed because of the exact arrangement of all the centromeres in a plate-like structure in the equatorial region, that is why we call it as an equatorial plate. And here in this one, the arms are freely present in the cytoplasm. They are not binded to the spindle fibers, only the centromere get binded to the spindle fibers. Likewise, during the metaphase, so we can see the perfect rod shaped chromosomes, and because of this attachment, so here this is the best stage to study the morphology. The metaphase is the best stage to study the structural morphology of chromosomes and also the shape of the chromosomes, or it is better to say it is the best stage to study the karyotype of the chromosomes. So, these are changes which are taking place during the metaphase of mitotic division. The third stage in the mitotic stage is mitosis is anaphase. Anaphase is considered to be the most active stage, and before anaphase, here as we are seeing that during the metaphase all the chromosomes are organized on the equatorial region, that is all of them are moving to the equatorial plate or equatorial region, and they are arranged on the equatorial plate. So, that process of all chromosomes coming to the equatorial region is called as a congregation. That congregation is taking place during the metaphase stage, and during the anaphase stage, as it is considered to be the one of the active stages of mitotic division, where the spindle fibers start contracting. When they contract, the pressure will be exerted on the centromeres of the chromosomes. As a pressure is exerted, the centromere will split. So when the centromere split, so if you see the diagram here, when the centromere split, each of the chromatid will be separated from each other, and each of the chromatid will acquire little bit of the centromere, and it will be acting as an independent chromosome. So here the daughter chromosomes will be formed. Likewise, the chromosomes are moving towards the opposite poles, with each of the chromosome is having one centromere. So this is centromere of the daughter chromosome. So this is a chromatid of the daughter chromosome. So these two are combinedly forming into a one single daughter chromosome. So here the spindles are contracting, that is these spindles will pull the chromosome towards this side, and these spindle fibers will contract and pull the chromosomes toward this side. So because of that pressure, the centromere will split, and each of the daughter chromosome will be separated opposite side. So here for this process they require the energy pool. So all that energy pool which is required for the movement of the chromosomes are formed during the interphase. So it is an active process. That's why I say that it is one of the active phases of the entire process of mitotic division. So during this process the daughter chromosomes are formed, and these daughter chromosomes are little bit having the contact with each other because of the interzonal fibers. They are connected with the interzonal fibers like this. The chromosomal fibers are separated from each other, but the interzonal fibers are connecting both the daughter chromosomes, and as the daughter chromosomes are moving apart, the interzonal fibers will be separated out, and here the group of daughter chromosomes will move towards the periphery, the one pole, and the other group will be moving towards the opposite pole. So at this stage each chromatid, as we know, it contains one DNA molecule. In the metaphase, during the metaphase, two chromatids are there with each chromosome. So each chromosome contains two DNA molecules there, but here during anaphase each chromosome contains one chromatid. Each chromosome or one chromosome contains one chromatid, one DNA molecule, and one centromere. Whereas during the metaphase, one chromosome contains two chromatids, two DNAs, but one centromere only. So that type of deviation difference can be observed between the metaphase and the anaphase stages. Next, last stage here, it is the telophase. Telophase is a stage where all the changes which are taking place during the phase are opposite to that of the changes that had taken place during the prophase stage. So here the viscosity of the cytoplasm will decrease. And the decondensation of the chromosomes will take place. Decondensation of the chromosomes, that is uncoiling of the chromosomes will take place. So because of that one, the chromosomes are converted into chromatin materials. So as like that of the interphase, they are once again converted into the chromatin materials. The endoplasmic reticulum and Golgi complex or Golgi apparatus, both of them have been reformed. Then the nuclear membrane and the nucleolus are reformed. So how does the nucleolus is formed? Means the chromosomes contain nucleolar organizer, that is a secondary constriction which can produce the nucleolus. So, some of the nucleoli are formed from the nucleolar organizer of the chromosomes. Likewise, the reorganization is taking place. And how does the nuclear membrane is formed? Nuclear membrane is formed from somewhat from the endoplasmic reticulum. So, likewise the changes that have been taken place exactly opposite to that of the changes that have been taken place during the prophase stage. So, here by the end of this one, two daughter nuclei are formed in the cell. So, if you see the diagram related to the telophase, so a daughter nuclei is formed at each pole like this. The nuclear membrane, nucleolus, and the chromatin. Little bit chromatin like structures can be observed here. So, this type of reorganization is taking place during the telophase. So, these two are called as a daughter chromosome. Sorry, these two are called as a daughter nuclei. So, whatever the chromosome content, the number of chromosomes present in each of the daughter chromosome will be same, and they are not only equal, but they are equal to the chromosomes that are present in the parent cell. And the ploidy of the chromosomes which are present in the nucleus are also exactly the same ploidy as like that of its parent chromosome. And here there is no change in the sequence of genes also, so that genetically and structurally, morphologically it is similar to the parental cells or parental nuclei. Okay, these are the 4 stages of karyokinesis where the nucleus have been divided to form into 2 daughter nuclei. Then after completion of this one, so the cell will enter into next stage called as cytokinesis, that is the division of cytoplasm, or the division of the protoplasm, or the division of the cell. So, here, so the division of the nucleus have been taken place during karyokinesis. Now, the division of the cell will take place in the plant cells by 2 methods. One is called as cleavage method and other is called as a cell plate method. In the cleavage method, first the constriction will start in the protoplasmic content. These constrictions which are formed on 2 opposite sides will move towards the center. That is a furrow-like cleavage. Start from periphery and move towards the center. So, this furrow-like structure is moving in the centripetal manner. So, after that, here the cytoplasmic content or the protoplasmic content is divided into 2 equal parts. Then the further whatever the furrow that is formed, cleavage that is formed. So, into this furrow like structure, so the hemicellulose, pectin and cellulose microfibrils, all these materials will fill into the furrow like structure that have been formed, which will solidify the thing, and it is converted into a complete cell wall like structure. So, this is a way how the cleavage method is taking place in the lower organisms, where the cell is divided into 2 daughter cells. And the other method of cytokinesis that occurs in the cells is cell plate method. So, here during the cell plate method, so we know that the karyokinesis have been taken place. So, 2 daughter nuclei are present inside the cell. Two daughter nuclei are present. Here the spindle fibers which have been remained, the small fragments of spindle fibers will come and which are present on the equatorial region, they will get arranged in the equatorial structure. So this spindle fibers which have been organized in the equatorial region is forming into a structure called as phragmoplast. This phragmoplast is generally barrel shaped in its nature. Then into this phragmoplast, so as we know that as they are fiber like structures, some small gaps are also been left between them. So here into this one, the Golgi vacuoles will, or Golgi vesicles will come to the region of the phragmoplast, and these Golgi vacuoles, the membrane of the Golgi vacuoles will unite with each other, and like this structure will be formed by forming a fluid material in between them. The membrane of vesicles will unite to maintain a fluid in between them. So they will fuse to form a two membrane structure. Between them the fluid matter will be left out, and this fluid matter will gradually convert into a solid material, or it will get solidified, and that structure is called as a cell plate. And this cell plate that have been formed will start formation in the center, and it will move towards the periphery, and it will move towards the periphery and attach to the lateral wall of the parent cell. So the cell plate which is formed from the center will moving towards the periphery, so we call the centrifugal growth of the cell plate. Centrifugal growth of the cell plate. Then the cell plate will undergo some changes and converted into middle lamellum. Gradually the pectin, cellulose and hemicellulose materials get deposited on either side of the middle lamellum, so that the cell is now divided into two daughter cells. So as and when the cellulose, hemicellulose and pectin have been formed, so we call that as primary cell wall material. So by the formation of a primary cell wall material, we can consider that the cell have been divided into two daughter cells. So second cell wall is formed when the cell becomes matured or when it become older. So that is not the stage where the cell is divided, but as and when the primary cell wall is formed, we can consider that the cell have been divided into two daughter cells. And here whatever the cell organs which are present, so those are nearly equally distributed between the daughter cells. And after some time, when the cells, after the completion of the cytokinesis, these cells will enter into the next stage of cell cycle. After the completion of cytokinesis, now the daughter cells will enters into next cell cycle, where they start increasing their size and forming the new cell organelles, increasing the number of cell organelles, and in which manner they will increase the number of mitochondria, means the mitochondria and plastids. The number of these mitochondria and plastids are increased by a process called as fission. That is, both of them will undergo binary fission to increase their number. So proplastids are the structures which are giving rise to the plastids by increasing their shape, by increasing the size and structure. So likewise, these are the changes that are taking place during the entire process of cytokinesis. So in the plant cells, it occurs, the cytokinesis will occurs by cleavage method in the lower organisms, whereas in the higher organisms it occurs by the cell plate method. Significance of mitosis. So significance of mitosis, as we have discussed, that it is a stage, or it is a type of division which occurs in the somatic cells. So it will help in growth and development of the organism. So particularly the meristematic cells which are present in the apical meristems or in the form of apical meristems and intercalary meristems. So here these two meristems will increase in the length of the plant body, that is what proportionate increase in the thickness of the plant body also. So that is, they are helping the growth and development of the organisms. and this is particularly in the plants, that is apical meristems and intercalary meristems in the plants. So, here these in the unicellular organisms, so this cell division or mitosis is equal to the reproduction, that is when the cell is undergoing or as it is a unicellular organism, when the cell undergoes division means it is giving rise to the daughter organisms, so that it is equal to the reproduction or giving rise to the daughter cells. Third one, healing of wound. Generally, if any part of the plant body have been injured, so the cells become dead. The surrounding cells present around the wound, generally they start they will regain the ability of division. They regain the ability of division and they start dividing and redividing. So, the regaining the ability of division is called as de-differentiation process. So, like they will heal the wound. So, that is by mitotic division only the healing of the wound will be taking place. Mitotic division of the cells surrounding the wound. Undergo de-differentiation. De-differentiation means the permanent cells which will acquire the ability of division and start dividing, and they will replace the dead tissues present in the wound region. Regeneration of lost parts. If any part of the plant body have been lost, so that lost part have been regained by the division of the cells which are present in the retained body. Then conservation of genetic integrity, or we can say the conservation of the genes or conservation of the genetical nature of the particular plant species. That is, whatever the chromosome number, whatever the genetical genes which are present on the chromosomes or the DNA, the same similar type of the same type of genes will be retained by the organism because of the mitotic division. Likewise, these are various types of functions related to the mitosis, and of course mitosis will also help in the wear and tear mechanism also. These are the significances of mitosis which are helping in the growth and development and other metabolic activities or aspects of the plants. In the significances, maintenance of surface volume ratio. So, as we know that mitotic division is useful for growth and development. So, the surface volume ratio of the cells is maintained because of the mitotic type of division. Next one is nucleocytoplasmic ratio. So, generally the nucleocytoplasmic ratio is differing in the smaller cells and the larger cells. The smaller cells generally they have more nucleocytoplasmic ratio when compared to the larger cells. So, here when the cell is dividing, the size of the cell will decreases. So, because of that one the nucleocytoplasmic ratio will increases. So, gradually what happens in the meantime, the cell will increase in its size and the nucleocytoplasmic ratio is maintained as like that of its parental cell. So, likewise these are the two other functions related to the significance of mitosis.